My name is John Gordon. I'm a third-year biochemistry major, and today I will be talking about the engineering of high-oil plants and their response to biotic stress. Triacylglycerol, also known as TAG, is a common lipid that acts as an energy storage molecule and a precursor to the synthesis of other important compounds, but it can also be used to manufacture cooking oils and to synthesize biofuel. For these reasons, scientists have been looking for ways of increasing the production of TAG in plants. The genes Wrinkled-1 and biotin attachment domain containing have been identified as genes of interest because they are major regulators of fatty acid synthesis, where Wrinkled-1 acts as a transcription factor and BADC acts as a down-regulator of the biosynthetic pathway. In order to amplify this pathway and increase the production of TAG, the model organism Arabidopsis thaliana has been engineered to express the genes Wrinkled-1 overexpressor and BADC RNA interference to increase and decrease the expression of these genes respectively. The purpose of this project is to engineer plants using Wrinkled-1 overexpressor and BADC RNAi that efficiently produce higher amounts of TAG in leaf tissue and to test how they respond to biotic stress. I hypothesize that Wrinkled-1 overexpressor BADC RNAi double mutants will experience higher oil production in leaf tissue than the single mutants and that high oil plants are more vulnerable to biotic stress. Due to restricted lab access caused by COVID-19, this project was delayed since the initial steps required in-person lab work. As a result, much of this project is still in progress. RT-qPCR, the insect feeding assay, oil extraction quantification, and agrobacterium transformation are still in progress and have not been completed. Plants were selected after undergoing antibiotic selection using hygromycin and Basta. So it was known that these plants expressed Wrinkled-1 overexpressor and BADC RNAi, but it was unknown whether they were homozygous for these genes. Homozygosity is important because it allows for greater expression of Wrinkled-1 overexpressor and BADC RNAi. Segregation analysis was done by taking the offspring of these 48 plants and performing another antibiotic selection. If the plant is not homozygous, then it carries the allele that lacks antibiotic resistance, resulting in some of the offspring being killed by the antibiotics. If the plant is homozygous, then all of its offspring will survive. The images to the right show how this was done. The offspring was compared to controls lacking antibiotic resistance. L17-2 was found to be homozygous while L46-2 and L46-3 were not. 28 of the 48 plants were found to be homozygous for Wrinkled-1 overexpressor and BADC RNAi. Reverse transcription quantitative polymerase chain reaction will be used to measure the expression of Wrinkled-1 and BADC and Wrinkled-1 overexpressor BADC RNAi double mutants to observe how much Wrinkled-1 and BADC are being overexpressed and suppressed. mRNA will be extracted using the triazole RNA isolation method. An insect feeding assay will be used to observe how the Wrinkled-1 overexpressor BADC RNAi double mutants respond to biotic stress. Plants will be enclosed in a container with cabbage looper caterpillars for 7 days, giving the caterpillars enough time to feed on the plants. Afterwards, the caterpillars will be weighed and observable leaf damage will be used to assess how the plants respond to biotic stress. This is based off the idea that if the plants do not respond well to insect feeding, then the insects will feed more and weigh more as a result. High oil accumulation could make plants more vulnerable to biotic stress because higher oil density could encourage higher feeding. Heavier caterpillars accompanied by a higher observable damage could be indicative of this conclusion. Oil samples will be taken from the leaf tissue of Wrinkled-1 overexpressor BADC RNAi double mutants and quantified and compared to RT-qPCR results to observe the effect that the overexpression and repression of these genes has on oil production. Oil will be extracted using the hot isopropanol method, quantified using gas chromatography flame ionizing detector, and prepared for GCFID through transmethylation. Agrobacterium transformation will be used to introduce the gene oleosine-1 overexpressor to the Wrinkled-1 overexpressor BADC RNAi double mutants. Oleosine-1 is a protein responsible for stabilizing oil storage bodies in plants. This gene is being overexpressed with the purpose of increasing the plant's capacity to store the higher amounts of TAG that are being produced for use in future studies. Wrinkled-1 and BADC are known to be key regulators of oil biosynthesis in plants, but it is still unknown how their overexpression affects oil biosynthesis and the plant's response to stress. As previously stated, homozygous Wrinkled-1 overexpressor BADC RNAi double mutants have been identified for use in this study. Experiments are underway to characterize these plants. Results are expected to be available by the end of August. Funding for this project was provided by the Turing Summer Scholars Program, the MU Research Council Grant, and the National Science Foundation.